Good morning. This would be a session for postgraduate students. We will talk about history of neurohumoral transmission. This will include neurohumoral transmission as well as some history about receptors, because ideally that should be a part of neurohumoral transmission only. Remember, before 1894, what was known was that the transmission in nerves is electrical. This was the concept. This was known that this is electrical transmission in the central nervous system and in peripheral nervous system also. But this all started in 1894. George Oliver and Edward Albert Schaffer. We always call them Oliver and Schaffer. I will try to find out the full names of these scientists, but you can remember Oliver and Schaffer. As I always say, we should try to remember the spelling also, so I always call it Schaffer. That helps me reminding that it starts with S, Schaffer. Very important scientists. They demonstrated that extracts of adrenal glands produced marked increase in blood pressure. This established the physiological importance of adrenal medullary secretions. And this work laid the foundation for later understanding of sympathomimetic substances. Then in 1899 and 1901, they were not working together. One was Max Lewandowski and another was John Newport Langley. Very important scientist, we always find written J.N. Langley. John Newport Langley. This is controversial. Max Lewandowski or it is Cybulski Lewandowski. There is confusion about these two scientists because both of them are found in different places in neurohumoral transmission. It is believed that Napoleon Cybulski just did what Oliver had done. He prepared the extracts of adrenal gland, and this is Max Lewandowski who found out that the effect of this extract is like sympathetic stimulation. So there is some confusion about this, but Lewandowski is an important scientist and another is John Newport Langley. They independently observed similarity between injection of adrenal gland extract and sympathetic stimulation. This was a crucial conceptual advance because it shifted attention from nerves alone to the interaction between chemicals and target tissues. Now, this always creates confusion that what is the difference between what Oliver and Schaffer had done. They also did the same thing. They produced hypertension with adrenal extent, and Lewandowski and Langley, they also did the same thing. And so it is important to remember the differences in the works done because this was a gradual progress. That is why I try to differentiate their works. Oliver and Schaffer demonstrated that the adrenal gland releases a chemical substance into the bloodstream that produces profound physiological effects. Oliver prepared extracts of adrenal gland and Schaffer injected these extracts. So they were working together. This was one of the earliest demonstrations of chemical transmission through blood and the concept of internal secretion, that is what we say endocrinology. So this proved that. On the other hand, if we see what this actually is an important part of endocrinology, as I said. What these people did, Max Lewandowski observed that stimulation of sympathetic nerves produced effects similar to this extract of adrenal medulla. He proposed that the adrenal medulla secreted a substance resembling sympathetic nerve action. This was an early step towards the concept of chemical mediation of sympathetic activity. However, he did not formulate receptor theory. That was done by Langley. Langley actually improved the concept further and he called it receptive substance. We'll talk about it in receptor, so we'll not discuss it here. But he went a step further and he said that there are some places in the cell where these substances they bind. So Langley was ahead, and actually Langley is very important scientist in neurohumoral transmission, like Dale, Paul Ehrlich. Similarly, Langley is very important. This was followed in 1905, Thomas Renton Elliott, T.R. Elliott, we find. He said that sympathetic nerve impulses release minute amounts of an epinephrine-like substance. And what he said in apostrophe is, the junction of nerve and muscle might be stimulated chemically by liberation of a substance. At that time, this was a revolutionary idea. This was not accepted at that time. Then in 1907, Dixon observed the similarity in the effects of muscarine and responses to vagal stimulation, something that was done earlier by Langley and Lewandowski. Advanced the concept that vagus nerve liberated a muscarine-like substance that acted as a chemical transmitter of its impulses. In 1907 only, this we find more frequently, described the actions of acetylcholine and other choline esters. So this name we find more frequently, Reed Hunt. We don't find Dixon so frequently. But Dixon also gave this concept. This was in the same year. Then in 1910, George Barger and Henry Hallett Dale, we always find him Sir Henry Dale. This is Henry Hallett Dale, noted that the effects of sympathetic nerve stimulation were same as the injection of sympathomimetic primary amines than by that of epinephrine or other secondary amines. Remember, Sir Henry Dale, Paul Ehrlich, they were pharmaceutical chemists, and they had real good knowledge about the structure of the drugs. So he observed that the effect of sympathetic nerve stimulation resembles primary amines, and note the secondary. Epinephrine was actually the extract of adrenal gland. So they were comparing sympathetic stimulation with adrenal gland extract till now. Now Sir Henry Dale and George Barger, they propose that this effect is not exactly the same, and they found out that primary amines resemble more like sympathetic stimulation. This was actually established the structure activity relationship, that they found out there is a difference between the activity of primary amines and secondary amines. In 1914, Sir Henry Dale investigated the pharmacological properties of acetylcholine and other choline esters and distinguished its nicotine-like and muscarine-like actions. Said there are differences in the activity of acetylcholine at different places. Introduced the term parasympathomimetic because he was so overwhelmed to see the activity of vagal stimulation in different places and the activity of these acetylcholine given from outside. And so he said, he coined the term parasympathomimetic to characterize the effects of acetylcholine. Dale also noted the brief duration of action, and he also found out that there must be some esterase that causes the degradation of acetylcholine immediately after its release. Dale introduced the terms cholinergic, nerves using acetylcholine, adrenergic, nerves using adrenaline or noradrenaline, and these terms still are fundamental to pharmacology. Then in 1920, Walter Bradford Cannon, we always find the name Cannon, W.B. Cannon. He said that stimulation of sympathetic nerves, including hepatic sympathetic nerves, resulted in the release of a substance that produced effects resembling those of epinephrine. They proposed that this substance acted as the chemical mediator liberated at sympathetic neuroeffector junctions and named it sympathin. He called it sympathin. Cannon had said that sympathin closely resembled epinephrine in many pharmacological properties, but differed from epinephrine in certain important respects. Exactly the same thing that Sir Henry Dale had said, that epinephrine does not resemble exactly what sympathetic stimulation does. So he also, then this was a classical example in 1921 of Otto Loewi. This was the definitive proof. What he did, he connected two frog hearts. The one heart is perfused and the perfusate from this heart would go to the other heart. One was donor, another was receptor. So when he stimulated vagus nerve of first heart, after some time the second heart also bradycardia was produced. This was his classical experiment, proved that a substance was liberated from the first organ that slowed the rate of the second. This was conclusive proof that something is released in the blood that acts on the other heart. This was till now everything was being done by Dale. In 1914, in 20 also, so many things were being proposed, but this was conclusive. Loewi referred to this as vagus stuff, that is vagus substance or parasympathin. This was conclusive. In 25, Otto Loewi and E. Navratil, I couldn't find the full form of E. It is always E. Navratil. They presented evidence of its identity as acetylcholine. Loewi also discovered that an accelerator substance similar to epinephrine and called accelerant stuff. What he observed that in summer season, vagus nerve predominantly releases this accelerant stuff. So there was seasonal variation in the effect of vagus stimulation. Then Feldberg and Kreyer demonstrated in 1933 that the cardiac vagus substance also is acetylcholine in mammals also, because these experiments were done in frog. So this was done in mammals also in 1933, this was proved. And this was the culmination of all this. Otto Loewi and Henry Dale, they jointly received the Nobel Prize in Physiology or Medicine for discoveries related to chemical transmission of nerve impulses. Their work firmly established neurotransmission is predominantly chemical. Specific neurotransmitters are released from nerve terminals. Drugs can modify neurotransmission at multiple sites. This how actually, again I tried to find out what changed over a period. In 1910 only, if you see, Barger and Dale primarily worked on structure-activity relationship of sympathomimetic amines. Their major contribution was that certain amines can mimic sympathetic nerve stimulation. Did not conclusively prove that sympathetic nerves release a chemical transmitter. Rather, their work strengthened Elliott's earlier hypothesis that sympathetic transmission may be chemical in nature. At this stage, the actual sympathetic neurotransmitter was still unknown. Researchers only knew that some chemical produced sympathetic-like effects. So in 1910, Dale did all these things. Walter Bradford Cannon, that we saw, went a step further by proposing that sympathetic nerves themselves release a chemical mediator at nerve endings. More specific idea than the work of Barger and Dale. Cannon's contribution included demonstrating release of an epinephrine-like substance during sympathetic stimulation and proving this substance is the actual neurotransmitter, naming it sympathin. So this was done by Cannon. And ultimately in 1946, von Euler found that the sympathomimetic substance in highly purified extracts of bovine splenic nerve resembles norepinephrine by all criteria used. This clarified that Cannon's sympathin was essentially norepinephrine and not epinephrine. So this was concluded in 1946, even after the Nobel Prize had been given in 1936. We now know that norepinephrine is the predominant sympathomimetic substance in the postganglionic sympathetic nerves of mammals and is the adrenergic mediator liberated by the stimulation. Norepinephrine, its immediate precursor dopamine and its N-methylated derivative epinephrine also are neurotransmitters in the CNS. This is all known now. Acetylcholine, in addition to its role as a transmitter, it has other roles like preganglionic fibers of both the sympathetic and the parasympathetic systems. They are cholinergic, motor nerves to skeletal muscles, and certain neurons within the CNS. They all use acetylcholine. These are major investigators. If you see acetylcholine, Dale and Loewe both worked on that. Noradrenaline, Ulf von Euler in 1946. We call it von Euler, basically full name is Ulf von Euler, sympathetic neurotransmitter. Dopamine, Arvid Carlson. Serotonin, Rapport and Page. GABA, Roberts and Frankel. Substance P, von Euler and Gaddum. They are responsible for substance P. And this is chronological. 1894 Oliver and Schaeffer, 1899 Lewandowski, 1901 Langley, 1904 T.R. Eliot, 1914 Henry Dale, 21 Otto Loewe, and 36 Lane and Loewe Nobel Prize. So at least these many things should be remembered. Then receptors also is a part of neurohumoral transmission. Before the 19th century, drugs action was poorly understood. Most explanations were empirical and based on gross physiological observations. Like drugs were believed to act by altering tissue vitality, modifying blood composition, or producing non-specific effects. No specific molecular target was known. In 1850s, Claude Bernard made one of the earliest important observations leading towards receptor theory. While studying curare, Bernard demonstrated that curare does not affect muscle directly. Instead, it acts at the neuromuscular junction. This established the principle that drugs act at specific sites rather than diffusing throughout the tissues. And Bernard's work introduced the idea of selective localization of drug action. This is the concept, selective localization of drug action. 1878 to 1905, John Newport Langley, we had talked about, considered one of the true founders of receptor theory. While studying nicotine, pilocarpine, atropine and autonomic transmission, Langley observed certain drugs stimulated tissues, others blocked the same responses, and These interactions occurred at specialized cellular sites. He proposed the existence of receptive substances, hypothetical components within cells that interacted with chemicals. So this was originally given by Langley. Langley suggested that nicotine and curare compete for the same receptive substance. Drugs act by combining with these receptive sites. Physiological responses depend on this interaction. This was the first clear formulation of the receptor concept. 1897 to 1908, Paul Ehrlich, another, this scientist we always find in antimicrobials, but his great contribution is towards receptor theory, because from this receptor theory only he concluded that if the drugs can bind selectively, then they can also kill selectively. This was his original theory. So he developed idea relevant to receptor theory, side chain theory. Cells possess chemical side chains capable of binding toxins. Toxins act only after binding to specific cellular components. He coined the famous statement, corpora non agunt nisi fixata. This is a very famous saying. Corpora non agunt nisi fixata means substances do not act unless bound. This principle became fundamental to receptor pharmacology. 26 to 37, Alfred Joseph Clarke, occupation theory. Drug's effect is proportional to the number of occupied receptors. Drug receptor interaction follows the law of mass action. He quantified the action. This introduced mathematical pharmacology into receptor science. The basic equilibrium concept is D plus R leads to DR. Clarke's work formed the basis of modern dose response analysis. DR we know, drug plus receptor is drug receptor complex. In 1930, John Henry Goddum, we always find his name with antagonist, expanded receptor theory by studying antagonism. He demonstrated that agonist and antagonist compete for receptors. Antagonism obeys quantitative principles. This allowed pharmacologists to calculate affinity, measure receptor blockade, and quantify drug potency. So Goddum is another name. In 1948, Raymond P. Alquist proposed alpha and beta adrenergic receptors. Alquist demonstrated not all adrenergic effects are mediated by the same receptor. Tissues contain distinct receptor subtypes. This was revolutionary because receptors could now be classified pharmacologically. Receptor heterogeneity became established. His work laid the foundation for beta blockers, selective adrenergic agonist, modern autonomic pharmacology we can call it. So Alquist in another name. 56. Everhardus Jacobus Arians and Robert P. Stephenson, another important names, recognize limitations of occupancy theory. Stephenson proposed that, so Arians and Stephenson, they were working differently, they were not working together. I have taken them together in the beginning because this is the same year, but their work was different. In Stephenson proposed that occupancy alone cannot explain maximal response. Drugs differ in their ability to activate receptors. This introduced intrinsic activity. Thus, full agonists, partial agonists, and antagonists could be explained mechanistically. Now, Arians, as I told you, also did almost the same thing, but there's a difference between what they did. Like in case of Langley, I told you Langley went ahead. compared to Lewandowski. Here also, Arians proposed that a drug may bind strongly to a receptor, high affinity, yet produce weak activation. Thus, he separated affinity and intrinsic activity. Stephenson further refined by proposing that response is not directly proportional to receptor occupancy. Agonists differ in their ability to generate a stimulus after binding. He introduced the concept efficacy. We always read affinity, intrinsic activity and efficacy. Affinity, intrinsic activity and efficacy. So these two things were proved by Arians, and Stephenson stressed more on efficacy. This concept later became more influential in modern receptor pharmacology. So you will find everywhere Stephenson. Arians is gone. Discovery of receptors as physical molecules. Remember, till now we are doing everything and we behaving like we know everything about receptor. But initially receptors were hypothetical entities. The major breakthrough came with radioligand binding studies, biochemical isolation, molecular biology technique, and this was in 60s, 70s. Researchers demonstrated receptors are real macromolecules. Now, in the definition we say they are macromolecules. Many are membrane proteins. Examples include all of them. Now we know it, but this is a very recent phenomenon. In 60s, 70s, we came to know about it. Second messenger era, 1950s, Earl Wilbur Sutherland Jr. discovered cyclic AMP. This led to second messenger concept. Receptors were now understood to activate intracellular signaling pathways, amplify cellular responses. Examples: CAMP, IP3, DAG, calcium signaling. This transformed receptor pharmacology from simple binding theory into signal transduction biology. Molecular pharmacology and receptor cloning. 80s onwards, advances in molecular biology led to cloning of receptor genes, receptor subtype identification, receptor sequencing. Major receptor superfamilies were characterized: G protein coupled receptors, ligand gated ion channels, enzyme linked receptors, and nuclear receptors. Modern pharmacology became increasingly molecular. Modern concepts in receptor pharmacology, now we see all these concepts: inverse agonist, biased agonism, receptor dimerization, allosteric modulation, receptor desensitization and trafficking. All these concepts are evolving now. Receptors are now viewed as dynamic signaling complexes rather than static binding sites. You can remember this starting from 1850s, Claude Bernard, whose specific site of curare action. 1878 to 1905, Langley, receptive substance theory. 97 to 1908, Ehrlich, side chain theory. 1904, Elliott, chemical neurotransmission hypothesis. 2637, A.J. Clark, occupancy theory. 30s, Gaddum, competitive antagonism. 48, Alquist, alpha and beta receptors. 50s, Stephenson, intrinsic activity. And 50s, Sutherland, second messengers. So these many things you have to remember in neurohumoral transmission. In the next lecture, we will start with various types of nervous system.